Welcome to Clot Chat. Today we're joined by Harriet from Queen Mary in London, and she's going to talk to us about our research. So Harriet, what is it that you are studying at the moment?
So the project I'm working on at the moment is trying to understand how platelets change as they age within the circulation. So platelets live for approximately 10 days, and during that time we think their function changes. And at the moment we've shown that young platelets seem to be more hemostatic cells and that they actively respond to injury and form clots. And hopefully in the next few months I'm going to start looking at what the old platelets do and see if they potentially may have a role in inflammation or whether they are just becoming kind of decrepit and cleared from the circulation.
So as we know, research is funded by all sorts of organisations and charities normally. How's the project that you're currently working on being funded?
So my project is funded by the British Heart Foundation, which fund work looking at heart disease and thrombosis. And my work fits within this because platelets are central to thrombotic disease. So if we can understand more about how they work, we might be able to reduce the impact of them in thrombosis.
So why is it important for us to understand the differences? What's the potential impact of understanding the difference between new and old platelets?
So there are a number of diseases where we see altered platelet lifespan. So for example, diabetes and chronic kidney disease, the lifespan of a platelet is much shorter. It's around 5 to 7 days rather than the 10 days in a healthy person. And we don't know why that is the case. So if we can understand the natural aging process of a platelet, we might be able to then understand why there's accelerated turnover of platelets in disease.
That all sounds really interesting. So this is probably a silly question, but how do you tell the difference between an old platelet and a young platelet?
A good question. And there aren't actually any kind of standardized ways of doing it. The way we do it is, so we use RNA as an indicator of platelet age, because obviously platelets are anucleate, so they only have a very limited amount of RNA, and this degrades or is translated throughout their lifespan. So the youngest platelets will have high RNA content, and then this gradually declines as they age. So we use a dye called thiazole orange, which binds to nucleic acids, and then we use a cell sorter to separate this population. So we have RNA rich ones and RNA low ones, and we just call them young and old, and then we functionally characterize those two populations.
Cool. So you mentioned cell sorting there, which actually I don't think I would hazard to guess that quite a lot of the society members don't use or are unfamiliar with. But are there any other techniques that you use or you've used in your project that you think that might be of interest to our society members, or that you think we should be utilising a lot more?
Yeah, so I mean, the two main techniques I use are flow cytometry and microscopy, which I think most people are familiar with. I personally prefer microscopy because I just really like pretty pictures, essentially. But obviously it's way more laborious and time consuming than flow cytometry. So I think the majority of our work, we try and do kind of a global characterization using flow cytometry and then look in more detail once we've got an idea of what might be happening using microscopy.
So if I remember correctly, Harriet, haven't you used Ingenuity pathway software to like compare and look at interactions between certain components of platelets? Have you got much experience of that, and do you think that might be a useful tool for other society members in their research?
Yeah, so when we have looked at the young and old platelets, we did some proteomic analysis on them. So we looked at the protein expression profiles, and we were able to identify proteins that were significantly modulated between the two populations. And whilst they're interesting to look at individual proteins, it's more interesting to see what pathways all of those proteins might be affecting. So we used the software called Ingenuity Pathway Analysis, and it's quite simple to use, really. It's you feed in your proteins, or you can do it with eicosanoids as well, that are different between two populations, and it predicts increases and decreases in particular pathways, so based on the expression of the proteins. So with our work, we were able to show that old platelets had a predicted increase in apoptosis and necrosis due to the downregulation of certain proteins. And the way it works is you put your proteins in and then it scours all the literature on the internet to look at what those expression levels have been associated with in other cell lines. So some of it is largely irrelevant because it can come up with, for example, like in like a cancer cell line, which obviously isn't applicable to platelets, but you can kind of exclude those from your analysis. So it's a really useful tool as a kind of a prediction. And then we used it to further look at pathways that might be affected.
So maybe you could tell us a little bit about your journey and kind of like how you got into the PhD and kind of postdoc that you're doing now, and kind of maybe later on we'll talk about what your plans are for the future.
Yeah, of course. So I think I've had a bit of an unusual route to where I am now. So I did my undergrad at the University of East Anglia, and I just did biological sciences. And I really enjoyed it, but I didn't particularly know what I wanted to do. And when I was there, I was asked to apply for a PhD, but at the time I was like, I don't think that is what I should do. So I didn't. And I left uni without a job, not knowing what I wanted to do. And I came back to my parents in Sussex and just applied for temporary jobs whilst I had just had some time to think about what to do next. And I ended up working for a charity called Sightsavers, who they provide kind of glasses and training for opticians in Africa and Asia. And I worked on a project, kind of like project managing some of the, setting up a kind of portal to track the progress of the projects. And that was really interesting, but it made me realise that I actually really like science, and I didn't particularly want to work in an office. So I applied to do a master's in London at Queen Mary, and it was on like vascular inflammation. And so I got a funded place by the Medical Research Council to do that for a year. And during that time, I did a 8-month project in Tim Warner's lab, where I looked at platelets and zinc. And yeah, I really enjoyed that, really liked platelets. And I decided I wanted to do a PhD, but I kind of left it too late by that point to start the following year. So I finished my master's and I got a job, another temporary job, working at the Royal College of Anaesthetists in their research grant department. So I kind of just did admin, but I got to sit on the meetings for their grant applications, which was really interesting. And just quite nice to know what the process is behind the applications and see what people talk about in those meetings, because it's a baffling world. And then I got a job working for L'Oreal and UNESCO on the For Women in Science programme. So they fund 5 postdoctoral researchers every year. And it's kind of a fund that can be used on anything. So it can be used on childcare, going to conferences, like lab consumables, just to make their lives easier, basically. And that was really great. And I met loads of inspiring women during that time. And just hearing their stories was incredible. And then I was very lucky in that Tim found some money for me to come back and do a PhD. So I went and started my PhD in 2016 with him. And my project initially was looking at platelets in chronic kidney disease. But that didn't go very far because there wasn't much of a difference. So then I started looking at mitochondria and platelets, and then onto the aging process of them. And I finished in February this year, and was again lucky enough that Tim got some funding for a postdoc, so I got to stay to keep working on that.
So I was actually going to mention you've been incredibly successful this year. So you have obviously completed your PhD, but you've been awarded several fellowships, I believe. And I was wondering if you would be willing to tell us about the ones that you have been awarded, and also the process about how you went about finding them, applying for them, and then, you know, any tips, etc., for anyone who is listening.
Yeah. So this year I've been given, I guess, one grant and one fellowship. I got a research grant for supporting women in science from Cayman Chemical. So they offer travel bursaries for conferences, and I'd applied for one, I think at the end of 2019, to go to ISTH in Milan this year. And I was successful in getting that, but obviously it was cancelled. So I emailed them and told them that thank you, but I don't actually need it this year. And they were like, totally fine, totally understand that. But because we can't give those out this year, we've introduced this new grant. So if you'd like to apply for that, you can. So I did. It was really not a very hard application. It was just kind of a background on yourself and then like what you'd want to spend the money on. I don't know how many people applied. I don't know if it was very widely publicized, so but I was successful in getting that, so that's really exciting, because it's like my first, I mean, it's only a small grant, but it's my first one that is just like solely mine. And then maybe a month ago I was awarded an ISTH and EHA training fellowship. So next year, hopefully, I'm going to go to Harvard to work with Kelly for 2 months and learn to work with megakaryocytes, which I'm really excited about. It'll be really nice to go and experience a different lab, and I'm really interested in megakaryocytes. Basically, since we went to the Gordon conference in Texas, and I learned way more about them, I was like, that's what I'd like to do. Because I think it fits with the work I've done before. But it would be nice to have a project that's a bit more my own. So hopefully in Kelly's lab, I'll learn how to work with megakaryocytes, and then might be able to get some preliminary data to apply for a fellowship. And again, the application for that wasn't that hard. It was just a 2-page document about what you'd want the money for and like timescales. But very early on in my PhD, Tim said, if you don't apply for things, you're never going to get it. So my kind of mantra is apply for everything and then you're likely to get something. And that's what I do whenever I see a fellowship or a grant that I'm like, oh, I'm kind of eligible for it, I'm just going to apply. Because more often than not with the small ones, they don't take that long to do. I mean, I'm not going to do it with massive fellowships or anything, but you can normally do it in around an hour. So I'm like, the payoff is definitely worth it if you get it.
So we'll all be tapping you up for tips and, you know, kind of where the new pots of money are.
I actually started jotting down all the places I've got money from or places I've applied for, because I thought it would make a nice kind of blog or something for the early career working group.
Yes, so you mentioned that you'd like to write a blog for the working group, and you've recently joined the working group, which is one of the initiatives that our early career society committee members have established. So what made you want to apply for the working group, and what are you hoping to achieve as part of the working group?
So I mean, I've been a member of Platelet Society for a few years, and I've gone to like Platelet Society conferences, but other than that, I've not been terribly involved in any of the things going on. And I thought it would be really a nice opportunity to get more involved, firstly. And secondly, I think being from quite a small platelet group, it's just nice to have like expand your network and kind of like just know who you can talk to about various issues. Because during my PhD, there were times when it would have been really helpful to know who to talk to about an experiment based on their expertise, because as I said, there are 4 people in my lab, which is quite a small lab, whereas, you know, in Birmingham, Reading, the groups are much bigger. So there is kind of always somebody you can go to ask. So I think it would be really great to kind of establish kind of sort of a network online that people know what everybody's doing if they need people to go to. And I guess kind of like in the practical sense, that would be very beneficial for a lot of people. But also I think I'd really like to be involved in kind of setting up some kind of mentoring program, because I think a lot of people talk about mentoring and being a mentor or mentee, but it never really comes to much. And I think at the moment, like, I'm in a very transitional period between PhD and postdoc, and it's trying to figure out where I kind of sit is a bit of a weird one. So I think for me personally, I would love to have a mentor. So if I could be part of a group that puts that together, I think it would be really, really useful for a lot of people. And even just kind of like peer mentorship as well. It doesn't have to be professors and stuff that are your mentors. And I think for me personally, I would rather have somebody that's a mentor that's like maybe a few years ahead in a career than me, because I think that's more relatable.
Okay, thanks so much, Harriet. It's been really, really great chatting with you. And on behalf of the Platelet Society, thank you for joining us today for Clock Chat.
Thanks very much for having me. It's been really great to talk to you today.
Join us next time on Clock Chat.